Welcome to the last week of lecture. So, this is lecture 58. Today, I am going to discuss about protein ligand interactions. So, we will see some methods where I will talk about protein ligand interactions. So, you have already seen this, let me see. So, I have already seen the types of protein ligand interactions, right. So, we can have enzyme substrate interaction, we can have enzyme drug molecule interaction. So, this will be a inhibitor. We can have DNA transcription factor. So, this is a protein, this is a DNA, protein nucleic acid interaction. We can have protein-protein interaction where this is a protein which is an antigen and this is the antibody which is also another protein. And we can also have another type of protein-protein interaction where this is protein or a peptide which is a hormone and that binds to this membrane bound protein which is the receptor, right. So, all of these interactions can be studied using the techniques that I am going to discuss today. So, this is again a recapitulation of what we have seen. So, this is a protein, this is a ligand and you get protein ligand complex, and we can measure how tightly this ligand is bound to this protein using a number called the dissociation constant. So, K suffix D, that stands for the dissociation constant, and it is given by the concentration of the free protein multiplied by the concentration of the free ligand divided by the concentration of this protein ligand complex. So, you can see that this is concentration, this is concentration. So, concentration square divided by concentration. So, its unit will be in concentration. The free energy of binding is related to this Kd by this value. So, R is the universal gas constant, T is the temperature at which you are measuring the dissociation constant, and this is log, natural log of Kd. So, that gives you the delta G. So, pictorially we can see it like this, that you have the free energy of the protein, it is somewhere here. So, in this we are plotting all the different conformations or the molecules, and here we have the free energy along the y axis. So, the free energy of the free protein is this, free energy of the ligand just by itself is this, and when you mix both, the free energy of this total system where both protein and ligand are free is this. But once the ligand binds to the protein, the free energy decreases to this. So, since there is a decrease in free energy, the binding will be spontaneous. So, if the free energy does not decrease, then the binding will not be spontaneous. So, here we are talking about spontaneous reactions. Okay. The typical setup, experimental setup to determine Kd will look something like this, where we have a protein here in a flask and a ligand in a burette or a syringe or some device, right. So, drop wise we will add the ligand to the protein and somehow we will measure what is the concentration of the protein ligand complex that is formed, right. You can also reverse this. So, the ligand can be here and the protein can be here. So, you add the protein to the ligand and measure it, right. Now, when you do that at every point, so it will be called a titration point. So, we are doing a titration. So, at every titration point you measure the formation of the protein ligand complex. So, this complex is plotted here and this is the amount of ligand you have added or the ratio of the ligand to the protein, right. So, this will increase. So, as it increases you will get more and more bound protein ligand complex, right. So, more and more protein will bind to the ligand. So, the concentration of the complex will increase. Eventually, it will reach some saturation where even after adding ligand, no more protein will bind, and that you will achieve if all the protein is bound to the ligand, right. Okay. So, we want to determine this protein ligand concentration, right. However, this is not something that we know. What we know is the total ligand concentration that you have added to your experiment, the total protein concentration that is there in the burette to start with or the flask to start with, right. So, these two you know, right. But now, if we look at the equation of Kd, it depends on free protein, free ligand and the protein ligand complex, right. But we can say that the total protein concentration will be the summation of free protein plus bound protein, because the protein is present in one of the two states, either it is free or bound. So, the total concentration will be the P total. Similarly, the ligand is either free or bound. So, the total concentration will be L total, and this is something that we know and this is something that we know, right. So, we can replace in this equation the free protein by P total minus PL. So that is what we have done here. So we have replaced just P by P total minus PL, and we have replaced just L by L total minus PL. So L total minus PL, and in the denominator we have PL. So in this equation you have P total that you know, L total that you know, you do not know what is PL, and you do not know what is KD, right? Now if we multiply the terms and collect them together, we will get a quadratic equation for PL. So you can actually see that PL times PL, you will get a PL square term, right? So if you collect all that and then you solve the quadratic equation, you will get a solution like this, where at this position you will get plus and minus sign, right? Now if it is plus, then it means that the protein ligand concentration, this will be the summation of total protein plus total ligand plus KD divided by 2 plus something, right? So which is physically not possible, right? So your protein ligand concentration cannot exceed the total protein or the total ligand concentration. So you can throw out that solution. So this will be minus, and that is what is shown here. So PL is given by this, right? So now you can see this is your experimental data, right? You know L total, you know P total. So these two you can, and you are getting a measure of PL from something. So we will see that indirectly we can measure it from fluorescence or heat released or things like that, right? So you will get a plot like this, right? So for each of these plots, they will obey this equation. So we can do a numerical fitting where we can figure out what is KD. So typically that is what is done, that this type of data will be numerically fitted to this equation, where PL is on the y-axis and this P total and L total is something that you know. P total will be most probably constant, L total is something that you are increasing for each titration point, and then you fit for KD and you will get the KD value. Okay. So here are some of the parameters that you can keep track of when you are setting up or designing an experiment, right? So remember that KD has the unit of concentration. Okay. Lower KD value means tighter binding. So if KD is nanomolar, it is tighter binding. If KD is micromolar, it is weaker binding. If KD is millimolar, it is very weak binding, right? So the following ranges of KD values are observed. For very tight binding, you get nanomolar or less than nanomolar. Tight binding will be 10 nanomolar to 1 micromolar. Moderate is 1 micromolar to 100 micromolar, and weak will be more than 100 micromolar. So it goes into the millimolar range, right? Now to determine KD values in this range, your experiment should be sensitive in this particular range. So to get something that is less than 10 nanomolar, your experiment should be sensitive in the picomolar range. Picomolar means that it should be able to determine the concentration of the free ligand or the free protein or the ligand bound complex, whatever it is determining, that should go down in the picomolar range, right? So to get this tight binding, which is 10 nanomolar to 1 micromolar, it should go down to nanomolar range. So 1 nanomolar or even less, right? So roughly whatever KD value you are measuring, your sensitivity should be to a good degree at least 10-fold less. Okay, so 10-fold less amount you should be able to detect with good reliability, right? Now the experiment that you will design will use or will detect the protein ligand complex in the range of 0.1 kD. So that is why you have to go 10-fold less up to 100 kD, right? So there will be a almost 1000-fold range of titration that you will do, right? So you will have to determine the protein ligand complex in this particular range. So your experiment should be sensitive not only in the lower range but also in the higher range, right? Okay. So again, these are some examples of protein ligand complexes, right, where you see that enzyme binding to its substrate will be quite weak. It will be in the micromolar range, right? Small protein binding to a signaling protein binding to a target, so hormone or something else binding to a receptor will be in the micromolar range. DNA transcription factor, because transcription factors, they have to bind to these promoter sites with high specificity and affinity, right? So in that case, the binding constant is much tighter. So we get nanomolar or even it can go down to picomolar range. Inhibitors of proteins or enzymes, they are Ideally, we want them to be in the nanomolar range. If you get something in picomolar, that's even better. And biotin binding to avidin is one of the strongest known non-covalent interaction, which is in femtomolar. Okay. Right. So now let's look at some experiments. So the first one that we are going to talk about is fluorescence anisotropy. We have discussed a little bit about fluorescence in lecture 56, right? So here, let's say we have a ligand. So we want to look at a protein ligand interaction, and let's say our ligand is a small molecule. So this is a ligand and it fluoresces, maybe it has its own fluorescence, or you have attached some fluorescence tag to this ligand, right? Now, what we do is we excite this with a vertically polarized light. So the light is vertically polarized like this, okay, and then it will emit this light. So it will absorb and then it will emit, and that is what we are going to detect. However, before emission, this ligand, since it is small, so the ligand will tumble, but since it is small, it will tumble faster, right? So it will tumble in all different directions, and that will depolarize this light, right? So whatever the light that was polarized, because of this rotational or tumbling motion of the ligand, you will get, what you will detect in the emission is a depolarized light. So you can measure how much it is depolarized, right? Okay. Now let's say we add a protein. So we are doing a titration now. We add a little bit of protein. So some of the ligand will bind to the protein. Since the protein is a bigger molecule and the ligand is bound to the bigger molecule, this whole complex will tumble at a rate which is much slower than this. Okay. Now since this tumbling is slower, the depolarization that happens will be smaller. So here we get a depolarized emission, here we get somewhat polarized emission, right? And this degree of polarization or depolarization will depend on how much ligand is bound to the protein. So as you proceed with your titration, as more and more ligand will bind, you will get more and more polarized light compared to this, right? So we can measure the rate of or the amount of change between this depolarized versus this polarized light and get an estimate of how much ligand is bound to the protein. So once we do that, so we can measure that. So this is A, right? So something that we are measuring, and we can then also normalize it. So there will be a maximum value, there will be a minimum value. So we can normalize that using this, and then you can see that this is the basic equation that we have derived. So the same equation comes back, right? So you fit to this, and you know this L total, you know P total, you are also measuring all of these A values. So you fit for Kd and you can get your dissociation constant. So it works well. This type of experiment works very well for moderate affinity range, which is 0.1 micromolar to 100 micromolar. Okay. There is another very popular and widely used experiment which is called electrophoretic mobility shift assay. So we have already seen polyacrylamide gel electrophoresis, but we saw it under denaturing condition. So here we are going to use the same method, same gel electrophoresis, but under native conditions. And typically this experiment is used to study protein nucleic acid interactions. And as I mentioned a few minutes back, that protein nucleic acid interactions are very tight, right? So EMSA is something that can actually detect much tighter interaction. If we are using radio labeled isotopes, then it provides very high sensitivity up to 10 to the power minus 18 molar. So you can measure picomolar, femtomolar binding constants using this type of experiment. So instead of radio label, fluorescence or chemiluminescence or immunohistochemical detection can also be used, but they will have lower sensitivity compared to this. So using fluorescence, you can go up to picomolar sensitivity, which is good for most applications. Okay. So this is the typical setup of the experiment. So here we are going to do titration experiment, but instead of having one flask, we will have a series or range of tubes. So these are small tubes, 1.5 ml sample tubes, and what you have is the DNA. So this orange color is the DNA, right? So you add maybe 5 microliter or 2 microliter of your DNA, the same amount of DNA in all the tubes, right? And this DNA is either radio labeled or it is labeled with fluorescence tag, okay? Then you add varying concentration of your protein. So So there will be one tube where you will have no protein, and then you add some more protein, some more protein. So you keep on increasing the amount of protein, but you have to keep the total volume the same. So you make it up with buffer, right? So this gray is the buffer, right? So now you have the total volume is the same. The DNA gets diluted to the exact same amount. The protein gets diluted. So you will have less protein here, you will have more protein here. So that is what you have. That is your titration. And then there will be one tube where you will have only DNA and buffer made up to the same volume. And then you take these samples, load them onto a polyacrylamide gel, which is under native conditions. So you will not add any SDS or any other denaturant, and you will run it. So then you detect it. If it is a fluorescent tag, you use some instrument that can detect fluorescence, or if it's a radio label, then you detect that. So you will get an image like this. So again, you see that this is the first lane or the first sample where you had only DNA. So that is what you see. This is the free DNA. So that is how you can actually tell which is your free and which is your bound. Right? Nothing here. You see something here. So this is your free DNA. And then so here it's done in the reverse order. So in this case, the amount of protein was less and the protein concentration is increasing in this direction. So as protein concentration increases, you see that this free DNA is decreasing, right? The band is becoming smaller and smaller, and there is a new band that shows up. So this is the bound DNA. So the DNA bound to the protein molecule, and it shows up here. So this is the DNA bound protein complex, and since it is bigger, it runs slowly. So there is a native page. DNA is negatively charged molecule, so it will run towards the positive end. All the free DNA, they will run at the same speed, right? However, when the DNA is bound to the protein, the size increases, so it will run slower. This is the mobility shift. That is why it is called electrophoretic mobility shift assay, right? So since the DNA is binding to this protein, there is a shift in its mobility, it runs slower, and you can detect the bound DNA. If you get a gel which is good like this, you can quantify this, or it is much better to quantify the free DNA. So typically that is what is done. You quantify the free DNA in each lane and you divide it by the intensity in this lane. So you measure the intensity in each of these lanes and you divide it by this. So that is normalization, and that tells you the saturation, right? So if this was 0, then this by this will be 0. It means that 100% is bound. If this divided by this gives you 0.5, it means that 0.5 of the DNA is bound, right? So that gives you a saturation. So whatever you get, you measure, you subtract it from 1, you will get the saturation, how much of the DNA is bound to the protein, and that can be expressed in terms of this. So this is the total protein concentration and this is the dissociation constant, and you can see that it does not depend on the DNA concentration, it only depends on how much protein you have added in each titration point, right? So you can plot this saturation as a function of protein concentration and it will look something like this. And here you see that it looks like a sigmoid curve because we have plotted this in a log scale. So you should be aware of that, right? So this is not a sigmoid curve, so there is no allostery or cooperativity in this. It is just because we have plotted it in the log scale, it looks like this, okay? So now we can fit this equation. So we have the theta, we have our protein total protein concentration for each of these titration point. So we can fit them and we can get the Kd value. So roughly you can see that when my protein concentration equals to Kd, theta becomes 0.5, right? So if I go to 0.5 like this, and if I go to this, so whatever protein concentration is here, that will be my Kd. So here the Kd will be somewhere in the range of 10. So if this is nanomolar, then this will be maybe 15 or 20 nanomolar binding constant. Okay. So what are the advantages and disadvantages of this experiment? So EMSA or electrophoretic mobility shift assay is a very simple, robust experiment, and it can be performed under wide range of conditions, right? It is very sensitive. It requires a very small amount of protein, so you do not have to make or purify large quantities of protein, or you have to use very costly ligands, right. So, since you are using very small amount, it is relatively cheaper and you can repeat your experiments multiple times to get good statistics. It is applicable to from small to large protein nucleic acid complexes, right. And then for relatively weaker binding affinities, fluorescently labeled nucleic acids can be used, right. So, if your affinity is something in the micromolar range or nanomolar range, you can use fluorescently labeled nucleic acid. Disadvantage, it requires considerable expertise and optimization. So, but this is true for any experiment, right. So, you have to do these experiments several times to get a good handle on it, and you have to optimize it according to your sample. So, there is of course that time that is taken. However, most biochemistry labs run gels. So, running a gel is something that students normally know. So, this is something that they can easily do or optimize. Dissociation during electrophoresis can make detection of complexes difficult. So, in many cases you will not be able to see the complexes well because of dissociation the band that is formed by the complex becomes diffused, right. So, you cannot quantify it, but then the free ligand or the free DNA band will be quite narrow and crisp, which you can easily quantify, and normally people use that for measuring the Kd value. No information is obtained regarding the binding site. So, you can tell whether your DNA is binding to the protein or not, and you can measure how tightly it is binding, but you cannot tell where it is binding, what is the binding site, and you will also not know the kinetics from this particular experiment, the way we have set it up. Okay. So, another very useful and very popular experiment that is used to determine Kd is isothermal titration calorimetry, right. And the instrument looks something like this. So, it has this thermally isolated chamber and there are two vessels, right. So, this will be normally metallic vessels. So, it can be gold or it can be some other alloy which is very inert. So, most of the samples can be easily used with them, and they have to be of course conducting. There is a reference cell and then there is the reaction cell, right, and both are in the same isolated chamber, both have exactly same volume. So, in the reference cell you fill it with your buffer. So, if your protein is in, let us say, PBS buffer, you will add the same PBS buffer here, and in this case you will add the protein solution, right. Then there is this syringe which will have the ligand. So, this can be a small molecule, this can be another protein, this can be DNA, so anything. Now, you will add your ligand to this protein dropwise. The moment you add it, some of this ligand will bind, and this binding can result in change in heat. So, it can release heat or it can absorb heat, right. And this chamber, what it does is it will try to maintain the temperature. So, it will measure that change in heat, okay, and that change in heat is plotted here. So, as you inject, as the first injection happens, there is some release in heat. So, you will get a peak like this, and then the instrument will go back to its equilibrium. So, it will go back to this, right. And then after some time another injection happens. So, you will get another peak and then it will go back. Then you do another injection, there will be another peak and it will go back. And you can see that progressively this peak will get smaller and smaller and smaller. The area under the peak gives you the heat that is evolved or absorbed at that particular injection, right. Now, why it is getting smaller and smaller and smaller? Because the first time when you are injecting the ligand, all the protein molecules are free, right. So, you will get the maximum amount of binding in the first injection. In the second injection, there are quite a few protein molecules which are bound. So, the amount of protein molecules that are free is less. So, when you do the second injection where you are injecting the same amount of ligand, but you will see lesser amount of proteins that are getting bound, right. So, the heat will be less, and finally, you will reach saturation. So, only 3 are shown here, but typically we do around 20 injections. So, by the time you reach go beyond 15, you will see that this heat is very low. So, it has reached almost saturation. So, A typical ITC data will look something like this. So you can see that the first injection gives you a very bigger peak, and then slowly it becomes smaller and smaller, and then finally it flattens out, right? You can still see that there is a peak here, right? Even though it has flattened out, but the same amount of peak is there. The reason for this is the dilution effect of your ligand. So your ligand is very concentrated, right? You are adding, let's say, 2 micromolar, which is going into 200 microliter of the protein sample. So there is a 100-fold dilution, and that dilution will result in some heat change, and that is called the heat of dilution, right? So typically what you should do is you should do another experiment where you inject the ligand into the buffer, not the protein, right? There you will see peaks of this height, but there will be no curve like this. It will be flat like this. So that heat of dilution, you can do the same number of injections, and you can subtract that heat of dilution from this, and that will remove the heat of dilution. So only heat that you will get will come from the binding. So then you can measure the heat or integrate this area under each peak, and you can plot it here. So this is the protein ligand ratio. This is the heat per mole that is released for each injection, and your data will look something like this. Then we can fit it to standard equation, which will look very similar to what I have shown you before, and you can get the Kd from this. So it turns out that once you get the Kd, you know the delta G, right? This difference in heat from here to here, right? Or in other words, the heat that you get from the first injection, that gives you a measure of delta H. Right? The heat that you get from the first injection, of course, you have to subtract the heat of dilution, that will give you the delta H. So if you know delta G, which comes from Kd, if you know delta H, you can also measure delta S. So ITC not only gives you the Kd or the dissociation constant, it also gives you the thermodynamic parameters, which are the enthalpy and the entropy. Okay. So these are some of typical ITC curves, right, for different protein ligand interactions. So you can go through this paper to see the actual conditions and actual binding assays. If you see something like this, this tells you that the binding is very weak. Okay. On the other hand, if you see something like this, you see everything is binding, and then suddenly it reaches saturation, right? So when you see something like this, then the binding is very tight. Okay. So weak, very tight, and then these are intermediate. So you can see that this is some binding and this is some binding, right? So this is still sharper than this. So you can just by looking at it, you can tell that this will be the tightest binding, then it will be this, then it will be this, and this will be the weakest binding. And once you do the actual fitting and measure the Kd value, you will see that is indeed the case. Kd is less than 1 nanomolar, so it is very tight binding here. In this case, it is around 5 nanomolar. This is 40 nanomolar, and this is 3000. So it is quite weak. So it's going into the micromolar range. So as I said that you can not only get the Kd value, but you can also get the delta G, delta H and delta S. So enthalpy and entropy values also. So some of the statins, so these are different statins which bind to HMG-CoA reductase. Okay, so that's an enzyme. So these statins binding to this enzyme has been characterized or studied by ITC, and the delta G shown in blue, delta H shown in red, and delta S or T delta S shown in green, they are all shown here, right? So you can see that if you just look at the blue, you can see the blue is more or less similar, right? So this is tightest binding, this is weakest binding, but they are in the similar range, right? However, if you see the delta H and delta S, they vary a lot. So you see delta H, so this one for binding of this molecule to the enzyme, it is mostly driven by enthalpy. You see red is the biggest, right? On the other hand, for fluvastatin, there is no enthalpy. It is completely driven by entropy, right? So this binding of this molecule, which is this, to this enzyme, this is completely driven by entropy, whereas for this molecule it is driven to the more by enthalpy than by entropy, and then these three molecules are in between, right. So, this is something that is very interesting, and you can sort of correlate that whether enthalpy will contribute more or entropy will contribute more by looking at the polarity of this molecule. So, you can see that this molecule is more polar and it has more enthalpic contribution, whereas this molecule is less polar and it has very less enthalpic contribution. Okay. So, not only protein ligand interaction thermodynamics, we are also interested in kinetics. So, how do you measure kinetics? Again, this is a ligand binding to a protein. The rate at which it binds will be given by the on rate k on. The rate by which it dissociates will be given by k off, or the off rate, right. And we can look at the rate of the formation of this protein ligand complex like this. So, the rate of formation is given by the forward reaction. So, the on rate multiplied by the ligand concentration multiplied by the protein concentration, which is shown here. And this complex, the concentration is decreased or the complex is destroyed by this reverse reaction. So, it will be the off rate multiplied by the concentration of the protein ligand complex, off rate multiplied by the protein ligand complex, and since it results in reduction, there is a negative sign here. So, this minus this gives you the rate of formation of the protein ligand complex, right. Now, at equilibrium, what happens is that this protein ligand complex concentration will not change with time, which means that dPL dt will be equal to 0. So, if you set this left hand side equals to 0 and if you take the ratio, you will get that k off by k on will be equal to P times L divided by PL, right, which is nothing but the Kd. So, under equilibrium, the dissociation constant equals to the ratio of the off rate and the on rate. Now, if we look at the dimension or the units of these two rate constants, on the left hand side it is dPL dt. So, it is concentration over time. K on will be 1 by time times concentration. So, time to the power minus 1, concentration to the power minus 1. So, that you can easily figure out by doing a dimensional analysis. So, this is concentration over time, this is concentration, this is concentration. So, this will be 1 by time times concentration. Similarly, k off will be only 1 by time. So, time inverse. Okay. And then if you put this dimensions here, you will get the same thing back, same equation back. Okay. So, what is a typical experiment to determine the on and off rate for a protein ligand interaction? This is our guiding equation. So, we have the ligand. Okay. So, let me just go back and show you this. So, protein ligand concentration is this. You see that the protein ligand complex is destroyed by this off rate, right. So, this is the reverse reaction. The protein ligand complex is getting destroyed by the off rate and you are getting protein plus ligand, right. So, if we focus on this, then we can say that okay, the rate of ligand formation is nothing but the off rate multiplied by the concentration of the protein ligand complex, which is this. Now, we can replace PL by this L total minus L, right, because L total will be the free ligand plus the protein ligand complex. So, we can solve for this equation and we can get something like this, where the free ligand concentration depends on the total ligand concentration times 1 minus e to the power minus k off by t, right. So, this is the first order reaction and this is what you get. So, you can simply see that when time t equals to 0, e to the power 0 is 1, right. So, 1 minus 1 is 0. So, there is no free ligand, right. At time t equals to 0, you have everything as bound. When t is a very large number, then e to the power minus large number. So, this becomes 0, L becomes L total. So, all the ligand is free, which means that this has completely dissociated, right. And remember that we have set it up as a irreversible reaction, that there is no rebinding of the protein and the ligand. So, we can plot this as a function of time. If I plot this as a function of time, it will look something like this. So, if this is my experiment, So if I can measure the free ligand concentration as a function of time, I will get this, and if I can fit it, I can extract my K off value, right. So I can determine what is K off. Now these are some of the typical setup parameters, right, that you should be aware of. So let's say you are aware of the K off value, that if you have some idea that the K off is something like, let's say it is 0.2 per second. Okay. So inverse of that is called a time constant. So if it is K off is 0.2, time constant will be 5 seconds. Okay. So the question is, how long should you run your experiment and how often should you measure? So these are the two parameters that you have to typically set in an experiment. Okay. So if I, let's say, run the experiment for a time which equals to this t, which is 5 seconds, you will see that your ligand will have dissociated, or the ligand free ligand formation will be only 63%, right. So if you are fitting to this curve, the K off value that you will get will not be very good. If you wait for 2 times of this time constant, that is 10 seconds, this will be 86%, right. This is somewhat okay. If you go for 3, if you go for 4, here it is almost 98%, right, more than 98%. So typically you should wait for 4 time constants. So if you think that, okay, your K off rate is 0.2 per second, then you have to run this experiment for 20 seconds so that you can get a good curve like this and you can get a good fitting, right. And then how often should you collect? The rate of data collection should be at least 2 times t by 5. Okay. So what is t? T is 5 seconds. So t by 5 is 1 second multiplied by 2. So your data you should collect your data at every 2 seconds or faster, right. So faster data collection is better, but if it will of course increase the size of your data set, but normally that is not a big concern. So it will be limited by the way you are doing the experiment. You should collect the data at least up to 4t. So in this case, you should collect your data up to 20 seconds. So collect data every 2 seconds up to 20 seconds. So you will have 10 data points and you can fit it to get a very good estimate of this K off. Fast binding reactions require faster data collection. So this will completely depend on the instrumentation, whether you can collect data at a faster rate. Slow binding reactions will require long time to reach endpoint. So you have to, even though you will collect data at a slower rate, but you will have to collect data for a much longer period of time. So it will be something where stability of the experiment will be very important. Okay. So we just saw that EMSA we can use to determine the KD value. If we set it up properly, we can also use EMSA to determine the off rate, right. So let's say you have a DNA which binds to protein, the DNA will also dissociate. So you want to determine how fast the DNA is coming off, and typically it will be a very slow process because the off rate will be very less, because protein DNA interactions are typically quite tight. Okay. So let's say you are studying something like that. So this is my DNA which is fluorescently labeled or radio labeled. I add my protein, so I will get a protein DNA complex, right. So you give some time for this complex to be formed and reach equilibrium. Then what you do is you add the same DNA without the label. So if this is fluorescently labeled, there is no fluorescent label. If this is radio labeled, there is no radio label here. Now what will happen is that this DNA will compete with this DNA to bind the protein. So, and since we have added more of this DNA, so it is more in concentration or volume, whichever way you want to see it, this DNA is more, so it will outcompete this DNA. So as you watch over time, you will see that less and less amount of complex will be there because there is dissociation. So you have this in one tube. So let's say you wait for 5 seconds, you add it here. You wait for another 5 seconds, then you add it to the next length. You wait for another 5 seconds and then you add it to the third length, right. But in this case, you will keep doing that while your gel is running. So this is something that is you have to do for a kinetics experiment. So you have to be very careful, right. So you are adding your sample while the gel is running, right. So of course they will run differently because I am running them at a later point. right? So by the time I add this, this is already moved, okay? So that's why we see a pattern like this. But since we are adding them at different time points, initially they will be more complex. As we go here, they will be less and less complex because more of this protein DNA complex is being replaced. As we go from here to here, we will see less and less complex because more and more of this labeled DNA protein complex will be replaced by unlabeled DNA protein complex, and you will see more and more of the free DNA, right? So we can quantify this, or we can quantify this as a function of time, and we can get the off rate. So this is my free ligand. I have it as a function of time, so I can go back to the previous equation and fit it to get my off rate, right? So this is my free ligand. I can fit it to get my off rate. Okay. So this is the last experiment that I will discuss, which is called the surface plasmon resonance, right? So it typically has this flow channel. There is a gold layer, and then there is a sensor chip, right? And what we do is we immobilize our protein here, and then we pass the ligand through this flow channel, right? So as you pass the ligand, some of it will bind, okay? So you reach a equilibrium. Then you stop passing the ligand, you pass buffer. In that case, what will happen is that the ligand will wash out, and then you will see whatever changes happens. So what are the changes? We incident some light, and that light is reflected. So this is total internal reflection that happens, and this reflection depends on the mass that is attached to this sensor chip, okay? So it changes something which is called an evanescent wave, right? So we are not going to go into the details of the physics, but there is a change in the reflection of the light which depends on the amount that is attached here. So if the free protein is there, there will be some signal. If a ligand is bound to it, then the total mass is changed, that signal is changed, right? And that is something that we can detect, and it is called RU, response unit. So it's some arbitrary unit that we refer to. So this is your response unit. You start your experiment, more and more ligand bind, so you will see the response changes, and then it will reach saturation. Then you stop passing the ligand and you pass the buffer, flowing the buffer. As buffer flows, you will see that more and more of this bound ligand are going out, so your response will decrease and you will get a curve like this. So this is because of association, binding of the ligand to the protein, and this is because of dissociation, the release of the ligand from the protein, right? And we can do these experiments by passing different concentration of ligands. So if you add less concentration of ligand, your response will saturate to a lesser point. As you keep on increasing the ligand concentration, your saturation point will go up, right? And then of course you start running buffer, you will get the dissociation. So we can just focus on the dissociation, right? And we can just focus on the dissociation, and we can fit it to the same equation that we have seen before, right? And we can get an estimate of the off rate. Now, since you have the off rate, you can also measure the KD by doing experiments like this. So you have this series of experiments, you have these different responses, right? So you get this different response as a function of ligand concentration. So from there you can get the KD, right? So once you have the KD, once you have the off rate, you can plug in the off rate, you can plug in the KD, you can also get the on rate. The instruments themselves have these fitting routines, so they can also fit the association part of the curve to get the on rate. So there are many ways you can get the on and off rate and the KD from SPR experiments. Okay. So this is a single experiment which provides both thermodynamics, which is KD, and kinetics, which is off and on rate parameters, right? It can be used to measure a broad range of dissociation constant starting from 1 nanomolar to 10 micromolar. It can be used to measure very fast kinetics, right? From 10 to a minus 5 per second to 1 per second. It is applicable to protein-protein interactions, protein nucleic acid interactions as well as protein small molecule interactions. So, this is a very versatile technique just like ITC. So, for today's lecture you can follow some of these books, especially these first two books, Molecules of Life and Leninger's Principles of Biochemistry. Thank you.